Biology, part 243. Well, there are homologs and analogs. An identification key can be made by using almost any characteristics of the organisms concerned. Its only criterion of success is effectiveness in enabling the observer to make a correct identification. Taxonomists have to be more selective in their choice of distinguishing characteristics when devising a comprehensive classification scheme, choosing only those which are believed to reveal true relationships between the organisms. Characteristics of this nature are called the homologs, where a homolog is defined as any structure or biochemical feature which is shared between two or more organisms by the virtue of common ancestral link. Example of the homologs structures in vertebrates could be backbones features or a five-finger limb. The organisms may share characteristics which are not homologs. These cannot be used as a classification because they do not necessarily reflect common ancestry. Wings, for example, common to bats, birds, and insects, but the different types of wings are not homologs. Features such as these, which are shared unrelated organisms, are called the analogs, meaning that they have same functions, although their basic structure may be quite different. The development of such features is different groups is called the convergent evolutions. The taxonomist has to distinguish between the homologs and the analogs in order to group organisms together in such a way as to expose their evolutionary links. One method of achieving this is called the cladistics, as outlined in the next section. Well, cladistics is a method of taxonomy based on the construction of the group or clades, comprising of organisms which share unique homologs. Birds, for example, from a clade share unique homologs, feeders, and mammals from a clade because they possess mammary glands and suckle their young ones. Both birds and mammals belong to a larger clade. Vertebrates through their common possessions of the backbone. Surprisingly, fish do not form a clear-cut clade on their own because although they have many features in common, such as gills, scales, fin, tail, none of these features is unique to fish. The relationship of clades is to each other may be expressed in the branching of the diagram called a cladogram. The simple cladogram shown in the figure based on the list of the shade features is given in the table as well. The joints or the nodes of the branching system represent the shared homologs, as we can proceed from left to right to the group, or we can find it there and we can find a similarity. Clearly, it is possible to make more than one cladogram for a group of organisms like this, but the criterion for choosing one above the other is simple. The best cladogram provides the maximum possible number of shaded homologs. The first step in the cladistic classification is the construction of a list of the characteristics such as shown in the table. To be as effective as possible, we can find them there. The validity of the method of the classification depends upon the assumption that if two organisms share the homologs, they must be related. There are very good theoretical reasons for thinking that this is so, and also for assuming that the more homologs share, the more closely related two groups must be. The modes of the cladogram should therefore represent the common ancestry, the phylogeny, and the organisms, and the entire branching system should reflect their divergent evolutionary history. The taxonomists have often tried to express the phylogeny in the schemes of the grouping, but their attempts have sometimes failed because they have concentrated on the search of the common ancestral types in the fossil record. and they have sometimes failed. They have concentrated on the search of common ancestral types of the fossil record.